Alright, so we're going to begin looking at the second part of bacterial genetics. So we're going to talk about gene regulation. We're going to talk about means of horizontal gene exchange between bacteria, as well as we'll talk about mutations and very briefly discuss DNA repair. Alright, so the first thing we're going to talk about is the organization of genes in the bacterial chromosome. Alright, so unlike eukaryotic chromosome structure, right, bacteria have their genes organized in what we call operons. Alright, so basically think of it as your grouping of genes that have a related or similar function. Alright, they're involved in the same biochemical pathway, for instance. So it makes sense that all those genes get basically expressed if you're going to have that particular biochemical pathway running. Alright, in this case it's all clustered together, so it can all be highly controlled and regulated. It can be turned off, it can be turned off, or turned on and turned off. Alright, so this is unlike talking about eukaryotic chromosome structure where genes are found on various chromosomes and their means of regulation is a little bit different. Okay, so here you're looking at an operon. Alright, so you have in your operon you have a control region, alright, here, which is going to be upstream of your structural genes. Alright, so these are going to be the actual product of this pathway of gene expression. Is you're going to be producing protein Z, protein Y, protein A. Alright, and all that's going to be controlled by the promoter and the operator. Alright, so let's apply this idea of an operon to one example in terms of bacterial cells. Alright, so I'm going to continue to go over the lac operon over the next series of slides. Alright, so this is a very important concept that you need to grasp, is how the lac operon functions. Alright, you need to understand how it's being turned on, how it's being turned off. Okay, so here you're looking at the lac operon. Notice in this graph, all right, it says induction. All right, so the lac operon is an induced operon. In other words, you have to induce it or turn it on for it to become active. All right, so it's kind of like, you know, your light bulb in a room isn't going to be on unless you flip on the light switch. Alright, that's what's going on here. You're inducing a change in expression. Well, in this case, it's the lac operon. Alright, so this requires that lactose be present in the media or in the environment, and the bacterial cell is taking up the lactose. Alright, so here you're looking at the metabolism of lactose. Alright, so you're looking at a series of genes that are involved in the catabolism of lactose. Alright, so here if you look at the graph, you have the relative increase on the y-axis, and on the x-axis you have your particular sugar that's being added. Alright, so in this case you have cell number that's increasing, you have total protein that's increasing. Alright, that's going on in the media, in the bacterial cells, alright, even before anything's added. Alright, and as soon as the lactose is added, you have a relative increase in the expression of a very specific protein. Alright, in this case, beta-galactosidase, alright, which is this little guy right here. LAC-Z gene, alright, it's beta-galactosidase. Alright, so this is an induced operon. All right, so in a normal setting, okay, your little repressor protein is bound to the operator. All right, and the operator is basically controlling whether or not RNA polymerase can transcribe the genes involved in lactose catabolism. All right, so if the repressor is bound, then transcription is blocked, and you don't have any of the proteins produced that break down lactose. All right, so usually this is going to be the case because lactose is probably going to be pretty rare in the environment. All right, so it doesn't make any sense to produce a protein or a series of proteins in a biochemical pathway to have a biochemical pathway turned on if that particular substrate isn't anywhere in the environment, if the bacterial cell isn't even taking it up. All right, it's like leaving a light switch on all throughout the day. You're wasting energy, you're wasting resources. All right, so what happens when lactose is present? Well, then what happens is you have a molecule, the lactose. The lactose will actually bind to the repressor molecule. And once bound, the repressor molecule, its shape is altered. All right, so it's no longer going to be able to bind to the operator. All right, so the operator is completely open. All right, so now RNA polymerase can go in and transcribe the genes needed for lactose catabolism. All right, so that's why you see here, once the lactose is added, the level of beta-galactosidase in the media increases. All right, because that particular pathway, that lac operon, gets turned on. All right, so you see that particular protein produced inside the cell. Now, you have other biochemical pathways that are actually repressed, repression pathways. All right, so in this case, you're looking at something like an anabolic process. All right, so it's like the synthesis of an amino acid. In this case, you're looking at arginine, for instance. All right, so cells are making a particular amino acid. All right, and so since amino acids are the foundational building block of your proteins, it makes sense that this particular operon is always on. Okay, so the cell is making arginine. Okay, so in this case, the repressor isn't bound. All right, RNA polymerase can go through and transcribe those genes that make up the biochemical pathway for synthesizing arginine. All right, so here in your graph, you have relative increase, you have cell number that's increasing linearly, you have total protein increasing linearly, you have arginine is being produced. All right, now it's being produced relatively at the same rate as the cell number and total proteins are increasing, so it's about the same slope. All right, so about the same rate of change. Now, once you add arginine to the media, what happens? Well, here again, the bacterial cell is coming into contact with arginine. All right, so it's going to take up arginine into the cell, and so it's a wasteful process to make something that's freely available in the environment. All right, it makes no sense for the cell to spend money buying food, basically, when it's a free buffet. All right, so that's what's taking place here. You have arginine being added to the media. Bacterial cell takes up the arginine, it's inside the cell, and this pathway for synthesizing arginine gets turned off. Okay, so the arginine binds to a repressor protein. That repressor protein binds to the operator. Now RNA polymerase is blocked from transcribing the genes involved in synthesizing arginine because it's in the environment. Okay, so that's why you see the level of increase of arginine flatline in terms of arginine biosynthesis enzymes, because arginine's there. You don't need to make any more. All right, it makes no sense to make something that's freely available. All right, so make sure that you can distinguish between induction and repression. Okay. All right, so this is kind of walking you through a little bit more in detail about the lac operon. All right, what's going on when the lac operon's off, and what's going on when the lac operon is on. All right, so you're going to have to be able to explain to me and describe what's going on when the lac operon is turned off, repressed, and when it's turned on. All right, so let's look here. You have your little I gene. The I gene is basically responsible for synthesizing the repressor protein. All right, that's a regulatory gene. All right, so that regulatory gene gets turned on and encodes for the active repressor protein. That repressor protein binds to your operator. All right, so now RNA polymerase can't transcribe the genes needed for breaking down lactose. All right, so what happens when it's turned on? Well, in this case, lactose will bind to your repressor protein. All right, now RNA polymerase can transcribe the genes in the lactose catabolic pathway because the repressor can no longer bind to the operator because there's an induced change in the structure of the protein. All right, so now you begin to produce the protein products of the lac-Z gene, lac-Y gene, and lac-A gene. All right, so you have beta-galactosidase, you have a permease, and a transacetylase. All right, and so all of these particular enzymes are involved in the breakdown of lactose into galactose and glucose. All right, because lactose is a disaccharide. All right, so what happens when all the lactose is used up? Well, in that case, it's no longer an environment. It can no longer bind to your repressor, and your repressor protein goes back and binds to the operator, basically turning off the pathway. All right, so kind of think of it as a timer, if you will, that you've preset. In this case, it's not really based on time, but it's based on the presence, absence, and amount of a particular substrate. All right, so what happens when you have other sugars present? All right, because lactose isn't the only sugar certain bacterial cells can use. All right, so one of the more readily available and usable substrates would be glucose. All right, so this is a little bit more involved than what I expect you to know, but I think it's important for you to understand that not all nutrient resources are metabolized equally. Okay, certain bacterial cells have a proclivity to using certain substrates over others. All right, think about it. If it's easier to break down one thing than something else, you're going to use that first thing, use that first nutrient resource first because it's the easiest to break down. All right. So here, if the cell has a lot of glucose in the media and very little lactose, all right, this lac operon is turned off. All right, the cell's using glucose. All right, it's going to preferentially use glucose. All right, now what if you have high concentrations of glucose and lactose? Well, think about it. Now your operator, some of your operators for this lac operon are going to be bound by lactose. Some aren't. All right, so this means that you're going to have this infrequent transcription of this gene sequence of the lac operon. Now, if lactose is high, then it's going to outcompete the glucose that's in the media, and you're going to have frequent transcription of the lac operon. All right, because your regulatory protein is bound by lactose, and so it can no longer bind to the operator. All right, so this pathway is constantly turned on in that case. All right, so this is something that we call a Diauxic growth curve. All right, so in a Diauxic growth curve, all right, you're looking at the utilization of two substrates. All right, so glucose is preferentially used first. All right, so in green, you're looking at basically the number of bacteria. All right, that's on the left y-axis. On the right y-axis, you're looking at the sugar concentration. All right, so in purple you have glucose, and pink you have lactose. All right, so notice that if you look at the curves of the bacterial growth numbers in green, the shape of that curve looks pretty similar to the shape of the curve of glucose as it's being broken down. All right, so they're almost mirror images of one another. As the glucose levels are going down at a constant rate, the bacterial cells are increasing in the media at a constant rate. All right, and as one rate increases, the other one decreases at the equal rate. Now, eventually, the cell will utilize all the glucose in the media, and so the bacterial cells kind of hit a plateau. All right, so they kind of hit this little stationary phase temporarily. All right, so they were an exponential growth phase, and now they're kind of leveling out. And notice that the level of lactose has remained constant all throughout this process, but now you have lactose present. All right, and this will be preferentially used. All right, so as the level of lactose decreases because the bacterial cells are utilizing it, bacterial numbers again begin to rise. Okay. All right, so it's a Diauxic growth curve. So it's showing you the preference of preferential use of one sugar versus another, and how it affects the growth rate and growth curve. All right, so again, this is just kind of going back over the structure of an operon. All right, so showing you the regulatory gene, the control region, the structural genes. All right, how operons can be turned on and turned off. All right, so you have a repressor protein. All right, that repressor protein binds to the operator region, then that operon's turned off. If you have a repressor protein that's not binding to the operator because it's been inactivated, then that operon's going to be turned on. All right, again, this is just kind of bringing you back to the lac operon. All right, so it's just many different figures showing you the same thing, one after another. All right, this kind of gives you a basic understanding of how this idea of induction and repression of operons occurs. right, so if you have a repressor protein, it can bind to your operator, which basically prevents RNA polymerase from transcribing these genes so they don't get produced. But then again, if you have a repressor and you have some type of metabolite that binds to your repressor, all right, like lactose, then your repressor protein won't bind to your operator. RNA polymerase can then transcribe all these genes into messenger RNA. That messenger RNA will then allow for the translation of those genes into functional proteins. All right, notice that these genes get transcribed together into functional protein products. All right, so it's this idea again of polycistronic messenger RNA. All right, you have many genes being transcribed at one time from one messenger RNA molecule. Again, also, you don't have any type of modification to your RNA sequence because there are very little to few introns in the bacterial chromosome. So there's no real editing process taking place. And all this takes place, again, in the cytosol of the bacterial cell. All right, so as a bacterial cell is transcribing messenger RNA from DNA, it can begin translating it into protein. All right, so it's a very efficient process. All right, so the question is, the lac operon, is it always on? Is it on when glucose levels are high and lactose levels are low? When glucose levels are low and sucrose levels are high? Or is it on when glucose levels are low and lactose levels are high? Or is it always off? All right, well, it's going to be on when glucose levels are low and lactose levels are high. All right, so your text goes over this topic of something called phase variation, but it does so very briefly. All right, so it's the idea that bacterial cells can turn on and off a certain set of genes or complement of genes. And as a result, it can lead to changes in a phenotype. All right, whether that phenotype is metabolic or the phenotype is the actual physical appearance of the bacterial cell. All right, so here you're looking at the same type of bacterial cell, just whether or not it produces a certain protein or not. So here you have things like pili. All right, so pili or fibrae. Your little fibrae here can be turned on or turned off. All right, so think about it. All right, these are structures that are sticking outside the bacterial cell. All right, so it's something that the immune system could potentially see. All right, so it's useful for attachment to surfaces and attachment to other types of bacterial cells or attachment to host cells, but the bacterial cell doesn't want to just kind of keep it sticking out there, willy-nilly, exposing it to anything and everything. All right, because if the immune system gets wind of it and sees it, well, then, it's going to be targeted for destruction. All right, so the bacterial cell can alter the expression of whether this set of genes responsible for synthesizing the fibrae is on or off. All right, so here you're seeing them on, below in the image you're seeing them turned off. All right, so this basically involves inverting a portion of the regulatory sequence in the DNA. All right, so here all the genes of interest are being transcribed in this direction. All right, so everything's good, everything's being turned on. All right, but you can take this portion here and you can invert it so it's going in the opposite direction, and basically this part of the pathway gets turned off. So as a result, this entire fibrae structure, the transcription of this gene here doesn't take place, right? Because this sequence is in the wrong orientation, right? It's inverted. All right, so as a result, your fimbriae don't get turned on at all, or don't get produced. All right, so hopefully you'll recall this idea of recombination. All right, you probably discussed it when you talked about the process of meiosis. You talked about genetics. You probably discussed recombinants, recombinant organisms that have recombinant genes or sets of genes. All right, so the process by which you get this kind of reshuffling and rearranging and of organisms or cells with different sets of genes. All right, one mechanism is this idea of recombination. All right, so here you have recombination events. All right, so bacterial cells can donate their genetic material to other bacterial cells. So there's different means of transfer. All right, but the end result is that you get bacterial cells that look completely different from the parent strain. All right, because they have new sets of genes, new combinations. All right, it's kind of like think of it as like the old Mr. Potato Head. Mr. Potato Head can have, you know, different sets of eyes and different noses and different mouths and different ears, and you can change his face. You have all these different types of combinations of things you could put together. All right, it's the same idea. You're reshuffling what you have in front of you, or if something pops up that's new, you can add that in and get new combinations. So you can get totally different new strains of bacterial cells. All right, so usually this involves things like plasmids. All right, so plasmids can be interchanged between bacterial cells. All right, so in this case, your plasmids are encoding for things like resistance to drugs and antibiotics, different types of disinfectants. It can enable the ability of the bacterial cell to either synthesize some type of new biological structure, or allow it to break down a new nutrient resource and use it for energy. You can have virulence factors, so like those fimbriae or bacterial toxins that can be found on plasmids. All right, they allow, in this case, the bacterial cell to establish itself in a host. As a result, promote an infection. All right, so looking at recombination. All right, so there's a little web link here, all right, for homologous recombination. You can click on that. It walks you through homologous recombination. It goes into a little bit more of the technical kind of mechanistic approach, but it's important that you just understand kind of the just main point of homologous recombination. Okay, so here you have a donor DNA. All right, you have a double strand break. All right, so that's what that endonuclease does. It cuts the DNA, one of the DNA strands, in half. All right, you get a double strand break in the DNA, and then you get single-stranded binding proteins. It's single-stranded DNA at that point in time. All right, at least a portion of it is. And here you have recipient DNA. All right, so this DNA that's new can invade the host DNA, and it can bind to a complementary sequence in the recipient DNA. Okay. And as a result, you have this kind of inter-exchange of DNA between the donor and the recipient. And you get this cross-link. You get this little X. Alright, so that this little crossover point, alright, your two strands of DNA, they can separate. Basically be cut. And you can end up with new recombinants. Alright, so you can get patches of genes. So you can get, or you can get donor here, you can get donor and recipient, get donor, recipient DNA, get recipient DNA, donor DNA, donor DNA. You get donor DNA, you can get recipient DNA. Alright, so homologous recombination allows the inter-exchange of an incorporation of DNA into host cell DNA, so long as it is complementary. Okay, so here you have you have media with bacterial cells. That are unable to synthesize tryptophan. Alright, it's a needed amino acid, and it's not available in the media. Alright, so when you plate these cells, nothing grows. Makes sense. Alright, if it's an essential amino acid, it's not available, they can't synthesize it, well, bacterial cells aren't going to grow. Okay, so what happens if you add DNA from bacterial cells that are able to synthesize tryptophan? Again, the media lacks tryptophan, but bacterial cells grow. Well, this is because the DNA from the tryptophan producing bacterial cells has been taken up into the tryptophan minus producing cells. It's recombined with the bacterial cell DNA, and as a result, you get recombinant bacterial cells that are able to produce tryptophan now. So now they're tryp plus cells. Alright, so all these recombinants become, alright, tryptophan producing cells, or tryp plus. Alright, so looking at mechanisms of horizontal gene transfer. Alright, so we'll discuss three major mechanisms. Alright, so this is going to be completely different from vertical transmission. Vertical transmission is from parent to offspring. Okay, so in this case, it'd be like going from parent bacterial cell to daughter bacterial cells directly through binary fission. Alright, that'd be vertical transmission. Alright, here we're talking about horizontal gene transfer between bacterial cells. Alright, it can be between bacterial cells of the same type or between bacterial cells of two different species. Alright, so, or even genera. Alright, so usually this involves the plasmids or parts of the nuclear bacterial chromosome. Alright, so here in this little short animation, you're seeing the exchange of plasmid DNA into this Streptococcus pneumoniae bacterial cell. Alright, so you're seeing horizontal gene transfer from one bacterial cell to another. Alright, notice one, here you have like E. coli, and the other one's Streptococcus, two different genera of bacteria, but you have horizontal gene transfer. All right, so the three different types of horizontal gene transfer are conjugation, transformation, transduction. All right, so conjugation. Conjugation is usually referred to as bacterial sex. All right, so bacteria are unable to have and multiply and reproduce by sexual reproduction, but conjugation's as close as they're going to get. All right, so you have two related species of bacteria. All right, one of these species of bacteria synthesizes a particular protein. All right, it's called a sex pilus. All right, so this sex pilus allows for the exchange of genes, plasmid DNA or chromosomal DNA, between the pilus producing cell and the non-pilus producing cell. All right, we'll talk about conjugation in the next couple of slides. You have transformation. Transformation is basically the uptake of naked DNA from the environment. All right, so bacterial cells just take up naked DNA into the host cell, and that naked DNA can incorporate itself through homologous recombination into the bacterial chromosome. You have transduction. Transduction is carried out by viruses or bacteriophages. All right, there's different types of transduction. All right, but in this case, you have DNA from one bacterial cell that's being transferred to another bacterial cell by that bacteriophage. All right, so we're going to walk through each of these types of horizontal gene transfer. All right, so this slide kind of gives you a nice overview of conjugation, transformation, transduction. Okay, so this kind of walks you through bacterial conjugation. Alright, so here, looking at conjugation. Alright, so we're talking about plasmid or chromosomal DNA. Alright, so first we'll talk about the exchange of plasmid. Now, this is a very specific plasmid. Alright, so you have something called a fertility factor that allows the synthesis of a pilus that's referred to as a sex pilus, or a conjugated pilus. It's a conjugation. Alright, the recipient cell is going to have a site on the surface of the cell that's going to be recognized by this sex pilus. Alright, so in this case, this fertility factor. The fertility factor is found on a plasmid. Alright, it's called an F plasmid. Alright, so you got an F. Alright, plasmid. Alright, this F plasmid has a fertility factor, and bacterial cells that have this F plasmid are referred to as F+. All right, bacterial cells that lack this plasmid and thus don't produce a sex pilus, quote unquote, sex pilus, are referred to as F- cells. All right, so what happens is you have this F pilus that's produced and extends out and attaches to the F- bacterial cell. All right, the F- cell is kind of drawn in and fuses. The membrane of the bacterial cell fuses with that of the F+ cell, and you have the exchange of part of the plasmid between the F+ and F- cell. All right, so you have the transfer of this plasmid from one bacterial cell to another. All right, so this is showing you a copy of the F plasmid. All right, so you have your origin of replication, you have your origin of transfer. All right, you have some other genes that are involved, like Tra region. All right, so that's basically used for transfer of the F plasmid to another, for assembly of this pilus structure, and you have some other gene sequences. All right, so here you have your F+ cell here. All right, notice that it has this really long pilus that it's producing. All right, so this is the sex pilus. All right, so here, this cell is your F- cell. Notice that it doesn't produce a pilus. All right, so what's going to happen is this F+ cell is going to exchange this F plasmid and transfer it to the F- cell. So the F- cell becomes a F+ cell. Right, so I'm not going to get to the differences between conjugation and gram-negative cells and conjugation and gram-positive cells. I'm just really interested in you knowing what conjugation is, knowing what F- and F+ cell is, and what's taking place between the two of them. I'm not going to really have you differentiate between processes that take place in gram-positive or gram-negative. All right, now the important thing here is that your F+ cell, all right, this is a conservative process. All right, so your F+ cell isn't going to become F-. All right, it's going to be F+, and it's going to always be F+. All right, it's only going to transfer a copy of the plasmid to the F- cell. Okay, so your F+ cell always retains a copy of the plasmid. All right, so here you have your donor F+ cell. All right, you have your pilus here, forms a little bridge. All right, so for a period of time, you have this interconnected membrane between your F+ and your recipient F- cell. Your F factor plasmid, all right, it's going to be replicated and transferred into your recipient cell, and eventually you end up with your F- cell that has a copy of the plasmid. All right, so this gives you a little bit more detail. All right, so you have your donor cell, your F+ cell, the F plasmid. You have your pilus interacting with the F- cell. You have the movement of your F- cell towards your F+ cell. The membranes fuse. All right, you have a nick in the F plasmid DNA, all right, and it gets transferred into your recipient cell here. And as it enters the recipient cell, it's going to begin to be replicated. And eventually, the two cells will separate. And your F- cell, initially becomes a F plus cell once it has this plasmid. Alright, so this F plasmid can have other traits on it, like antibiotic resistance, resistance to toxic metals, and other things like that. Alright, so this F plasmid is encoding for specifically this process of conjugation, the sex pills, but we can encode for other really cool traits as well. Now, you can have conjugation that involves a bacterial chromosome, alright? But the bacterial chromosome is really big, alright? So we have this process called high-frequency recombination, where the plasmid, the F plasmid from the donor cell, alright, when it transfers to a recipient cell, alright, it can actually incorporate itself, integrate into the bacterial cell nuclear DNA, like here. Alright, here in pink or purple, you have your nuclear bacterial chromosome. Here you have your F plasmid. Alright, so well, starting off, all right, here you have your you have your bacterial nuclear DNA, and as well as your F plasmid. Your F plasmid is actually incorporated into the nuclear DNA bacterial cell. All right, that begins to replicate and is transferred to your recipient cell. Now, only a partial copy of this plasmid gets transferred in. Alright, so now you have, along with the F plasmid genes, you have some genes from the host bacterial nuclear chromosome that get transferred to. And so that can incorporate into the chromosome of the host cell. Alright, so you can have the exchange of genes that are actually found in nuclear DNA, not just the plasmid. Alright, so this is high frequency recombination. Alright, because here the plasmid incorporates into the bacterial chromosome. And as this plasmid is cut out and it's replicated and copied and transferred into the F minus cell, right? Then it's going to not only get the plasmid, but it's also going to get other genes. And then this series of genes are going to incorporate into the bacterial nuclear chromosome. All right, so looking at this a little bit further, all right, so you have the F plasmid. This is kind of walking you through this process of what an HFR strain is. All right, so an F plus cell, all right, an F plus cell has a bacterial chromosome and the F plasmid, and they're completely separate. All right, now what can happen is that F plasmid can incorporate itself into the bacterial chromosome. At that point in time, your bacterial cell is labeled a HFR cell. All right, so your F plasmid encodes for genes, for instance, G. And you have in the nuclear DNA, you have A through F, for instance. All right, once it incorporates in, you have your little F plasmid with gene A, gene F, Fe, D, C, and B. Alright, so you have your HFR strain. Your HFR strain can interact with the F minus strain. Alright, so your HFR strain can still synthesize your sex pills. Alright, so your F plasmid that's inside the nuclear DNA will begin to replicate and be transferred into the F minus cell. Alright, now since it's nuclear DNA, alright, these bacterial cells are going to be held together very long through conjugation. Alright, they're going to separate. So only a portion of the F plasmid and the bacterial chromosome will actually enter the F minus cell. So as a result, alright, you're left with your HFR cell that you had initially. Alright, so this is your HFR. Here, it's still an F minus cell. Alright, the reason being is because it doesn't have the complete gene sequence of genes needed for synthesis of that F plasmid. But in this case, you have a portion of the F plasmid and you have a portion of the bacterial chromosome that can combine and align with the host cell chromosome in green here, for example. You have the recombination event and you get new genes. So this is an F minus recombinant. Alright, so let's take this cell here, give you another example. HFR cell, and you have an F prime cell. Alright, so what can happen is that this HFR cell, the plasmid itself, can actually excise itself from the bacterial chromosome, but when it excises itself, it's not a perfect process. Alright? So it can, when it removes itself from the bacterial chromosome, you get a plasmid with part of the bacterial chromosome on it. Alright, so here you end up with a bacterial chromosome lacking genes A and B, and you end up with the F plasmid with genes A and B from the bacterial chromosome. Now, at this point in time, this is referred to as a F prime cell. Alright. So this F prime cell can interact with the F minus cell. With this F plasmid, it gets transferred to your F minus cell. Alright, so now, as a result, you end up with a bacterial cell that has that becomes F prime, right? Because this plasmid gets exchanged here, all right? And as a result, you end up with a bacterial cell that has two copies of genes A and B. All right, because it has two copies has a copy in the plasmid and a copy in the host cell chromosome. All right, so this is what we refer to as marrow. All right, diploid. All right, so we generally think of bacterial cells as haploid. All right, so they only have one gene, one set of genes for a particular product in the chromosome. But in this case, you can end up with instances where you have a marrow diploid cell. All right, so it's partially diploid, but not fully. All right, so here, looking at this process of conjugation. All right, so you can have the transfer of resistance plasmids. All right, they encode for like antibiotic resistance, resistance to heavy metal toxicity like arsenic and antimony, mercury and lead, among other things. All right, all this is encoded in the plasmid. Alright, so transformation involves the process of taking up naked DNA. Alright, so you have a bacterial cell that dies. Alright, so it lyses open and it can release this chromosome. Alright, and it's plasmids as well. And this DNA can be taken up by another bacterial cell. Alright, so these fragments of DNA can be transferred from the environment, taken up from the environment into the bacterial cell. And then this DNA that's foreign can be incorporated into the bacterial chromosome. Now, bacterial cells that are capable of taking up naked DNA are referred to as being competent. Alright, so there are certain bacterial cells that are naturally competent. Alright, they do this all on their own. And there are other bacterial cells that are a little bit more finicky that you have to make competent. You kind of force them to become competent. Alright, you can do this chemically. You can do this using electricity and shock. Alright, so there's different methods of inducing competency and promoting transformation. Alright, so here you have a little bacterial cell and you have fragments of DNA in red in the environment. Alright, so these fragments of DNA are taken up by the bacterial cell. And where there is complementary base pairing between this naked DNA that you've just taken up and the host chromosome, you can have homologous recombination. Alright, so here you have A and B. Alright, so this portion here will actually switch out with this portion there. Alright, so the host AB genes swap out the AB genes from the naked DNA. Alright, so now you have a recombinant. And then all the other DNA that's foreign and naked gets broken down and degraded by enzymes. And so now this bacterial cell is a recombinant. Bacterial cell. Alright, so looking at the process of generalized transduction. Alright, so here in generalized transduction, right, you have your little bacteriophage that infects a bacterial cell. And in this case, as your virus is going through the process of synthesizing new viral DNA or RNA, right, depending on where, and synthesizing viral proteins, you can have some of the host cell DNA that's still left viable that isn't chomped up by enzymes, and this gets incorporated into the phage head in place of the bacterial phage DNA or RNA. All right, in this case, this little phage that now has the host cell DNA in it, when it leaves the bacterial cell and it lyses open, this phage then infects a new bacterial cell. Now, this phage is technically dead at this point, right? Because it's transferring bacterial DNA and not viral DNA. All right, so now this new DNA from the bacteria can combine with the host cell DNA through homologous recombination, and you get a recombinant bacterial cell. Alright, so this kind of walks you through the process again of the lytic and lysogenic cycle. So you may want to refresh yourself on the lytic and lysogenic life cycles of a virus, of a phage virus. Alright, so here you have your phage, it infects bacterial cell. You have your host bacterial DNA, it gets chomped up. Alright, now when you have your new phage head, it gets synthesized, you can have bacterial DNA that gets incorporated in place of viral DNA. So now you have your little bacterial virus with bacterial DNA in place of viral DNA. It infects the recipient cell. Bacterial DNA can combine through homologous recombination with the host cell chromosome, and you get a recipient cell that's now recombinant. It has its own DNA and DNA from some other bacterial cell. Alright, so specialized transduction involves a different scenario than generalized transduction. Alright, so in this case you have a very specific part of a host cell genome that gets incorporated into the virus. So instead of this random bacterial DNA fragment, you have a very specific part of the host cell genome that's incorporated into the virus. Alright, so In this case, you have a prophage. All right, so a prophage is basically a viral phage DNA that's incorporated into the host chromosome. All right, so that's a prophage, like here. That prophage will excise itself from the bacterial DNA. Now, it's not a perfect process. So in this case, when it excises itself, you get part of the bacterial DNA along with the viral DNA. All right, so you have your viral assembly. You have that viral DNA that gets incorporated into the phage, and that phage goes on to infect a new bacterial cell. And this viral DNA that also has bacterial DNA in it will recombine and integrate into the host bacterial chromosome. Alright, so this is one of your more temperate phages. Alright, one of your phages that undergoes the lysogenic, not the lysogenic, but the lysogenic life cycle. Alright, so it's not going to automatically begin replicating and rupture the bacterial cell. In this case, it actually incorporates itself into the bacterial cell chromosome. Alright, so here, looking at a little bit clearer example. Alright, so you have host cell DNA in light green. In dark green, you have phage DNA. And in pink, you have DNA, alright, it's for galactose genes. Alright, so this induction process of the phage DNA excising itself. Alright, you end up with this viral phage DNA along with the galactose genes attached to the viral DNA. Alright, so here you have your phage DNA in dark green. Alright, so it's a normal phage. It goes on to multiply, produce new phages, and little baby phages get produced that have normal viral DNA. Alright, well in this case, when you have the event where part of that galactose gene also gets cut out along with the viral DNA, alright, so you get viral DNA and bacterial DNA. Alright, you get your little viruses that get produced, and all of these viruses, alright, can transduce galactose genes to a new bacterial cell. Alright, so transposons are commonly referred to as jumping genes. Alright, so they can bounce around the host genome or bounce around between a host genome and a plasmid. Alright, so you're going to find transposons in both bacteria and in eukaryotic cells. Alright, so these things are able to replicate themselves. They can then jump to a new location. Or they can physically remove themselves from the host DNA and insert themselves somewhere else in a host DNA. All right, there's different types of transposons. We'll go over a couple of these types in the next couple of slides. All right, so think about it. You have this sequence of genome. It doesn't really encode for anything, but it can excise itself and insert itself elsewhere in the genome. So you can imagine that if it inserts itself in a gene, that it can inactivate that gene. So you could get a variety of different phenotypes as a result. You can have activation of genes, you can have activation or even overactivation of genes, all right, due to these transposons inserting themselves in different parts of the genome. All right, so here, what you're looking at is a transposon. All right, so what you have is, for example, in a plasmid, all right, you have an example of a transposon. And this transposon has a region of inverted repeats. All right, these inverted repeats, all right, are what are going to be homologous to the host cell chromosome or the plasmid. All right, now this transposon can encode for resistance genes, all right, along with the ability of this transposon to insert itself into the host cell chromosome or insert itself into a plasmid, as well as its ability to excise itself. All right, so this transposase gene is what allows it to basically insert itself and excise itself from the host cell genome. All right, so you have target sequence. All right, so you have one polypeptide of DNA. All right, one strand, all capital letters, light green. All right, and you have the complementary strand of DNA, capital letters, but they all have prime symbols after them. All right, so here you have your little transposable element that can insert itself into the DNA. All right, you can actually end up with a duplicated sequence of genes in this case sometimes. All right, so duplicate copies of genes. Now, brings up an interesting point. This means that genes can now specialize for certain things. You can end up with modifications to genes that the product of one gene, like gene B, let's say over here you have like B1, over here you have like B2. All right, well, the product of B1 may not be exactly like the product of B2. All right, so you can have specialization of genes. All right, so you have two different types of transposition. All right, so you have conservative transposition and replicative transposition. All right, so conservative transposition, all right, you have host cell DNA, you have a transposon, you have a target DNA molecule, and you have a target sequence. Well, in this case, your little bitty transposon will excise itself and attach itself to the target sequence in the DNA molecule. All right, so you could be going from host chromosomal DNA to a plasmid, or plasmid to host chromosomal DNA, or different parts of a plasmid or different parts of host cell DNA. All right. In replicative transposition, what happens is that transposon will actually duplicate itself. It remains in the host cell DNA in this example, but then also is going to be inserted, the replicate is going to be transferred to the target DNA molecule, and it inserts itself into the target sequence. All right, so transposons. You can have a transposon, so you have gene A, you have a transposon of gene A. It can insert itself into a chromosome, all right, through transposition. Here, it's inserting itself into gene 2. All right, so now gene 2 gets basically turned off, all right, because you have a portion of gene 2 here, you have a portion of gene 2 there, but you don't have the entire reading frame of that gene. It's cut off. All right, so that gene gets disrupted. All right, so that product of gene 2 turns to the protein doesn't get produced. All right, so which of the following mechanisms of horizontal gene transfer involve the transfer of bacterial DNA from a phage? All right, is it transposition, conjugation, transformation, or transduction? All right, in this case, it is transduction. All right, so looking at mutations. All right, we're going to talk about different types of mutations. All right, so you can have, in this case, nucleotide pair substitutions. All right, so in this case, you swap out one nucleotide, like for instance, an adenine for a guanine, for instance. All right, swap it out. All right, this is what we refer to as a point mutation. Now, mutations, real mutations occur in the DNA. All right, and real mutations have to be heritable. Okay, a mutation that's not heritable is not a true mutation. All right, mutations can be advantageous, or it can lead to the development of new traits, new abilities. All right, or more times than not, they're disadvantageous, right? Because they actually end up in the production of a protein that's non-functional. Or if you have a mutation in a region of the genome that encodes for a kind of a core process that cell needs to survive, well, that process is turned off, and the mutation isn't alleviated or repaired, then a cell is going to die. All right, so looking at different types of mutations, you have things like silent mutations. Silent mutations, you end up with the same amino acid that's encoded for. All right, so even if you change one of the nucleotides in the genome, all right, the protein, the amino acid so like amino acid 1, if amino acid 1 is lysine, let's say you alter that region of the DNA for that gene, but that point mutation results in the same amino acid, like lysine, again, this is a silent mutation. You're not going to notice it unless you're actually looking for it. A missense mutation, is where you alter the nucleotides in the gene. All right, you alter one of the nucleotides, and you end up with a different amino acid at that position. All right, so position 1, if amino acid 1 is lysine, and let's say you get instead an arginine. All right, it's a different amino acid. All right, that's a missense mutation. A nonsense mutation results in a truncated or shortened protein that's non-functional. And then a frame shift mutation results in a alteration in the reading frame. All right, usually this involves an insertion or a deletion of nucleotides. All right, so kind of walk you through each of these. All right, so a silent mutation. All right, so again, this is taking place in the DNA. All right, so here you have in a normal DNA template, you have methionine, lysine, phenylalanine, glycine. Okay, cool. For 4 amino acids. Let's say here, for this sequence, in place of a G, you have an A. Well, that means that your functional messenger RNA will have a uracil instead of a cytosine. All right, so you get a completely different nucleotide in place of it. But look at it, GGC and GGU. They encode for both glycine. All right, so the same amino acid. All right, so again, this ties back in with the redundancy of the genetic code. All right, this wobbly base here allows for most amino acids, you can change the third letter of the codon, and for most amino acids, you end up with the same amino acid. All right. Now, looking at a missense mutation. So here, instead of having a C here, you have a T. All right, so that means that your corresponding messenger RNA, instead of a G, will have a A. All right, instead of GGC, right, codes for glycine, you get AGC, which codes for serine, a totally different amino acid. All right, so this is a missense mutation. A nonsense mutation. All right, let's say that here, instead of a T, you have a A. All right, so in your RNA, you have, instead of an A, you have a U. Well, UAG encodes for stop codon. All right, so you only translate the first amino acid, methionine, and that's it. Because you have methionine, and the next codon is a stop. Well, you've shortened the protein sequence from 4 amino acids down to 1. All right, so this is a non-functional protein. It's a nonsense mutation because that protein can't do anything. All right, looking at insertions and deletions. All right, so let's say that here you insert an adenine. Well, that means that here in the RNA you'll have a uracil. All right, so now you have, instead of AAG for lysine, you have UAA, which is a stop codon. So again, you produce a shortened protein. Here, let's do a deletion. All right, so let's cut out this adenine, all right, which shifts the reading frame this way. All right. So now, when you're looking at your messenger RNA, you get, instead of UUU, you get UUG. UG encodes for leucine instead of phenylalanine. You get, instead of GGC, you get GCU, alanine instead of glycine. So you get a totally different sequence of amino acids when you start shifting the reading frame. Either you're adding nucleotides to it or you're removing them. You shift the reading frame and get new amino acids. Now, what happens if you delete 3 nucleotides? All right, well, here, let's delete TTC. All right, let's delete this sequence here. If you cut that out, right, you actually end up shortening the overall protein. You lose 1 amino acid. All right, methionine, phenylalanine, glycine. The reading frame shifts to the left. All right, but none of the amino acids you encoded for previously, or would have been encoded for, are changed. All right, so methionine, phenylalanine, glycine. You just cut out this amino acid. All right, so you have no frame shift, but you're missing one amino acid. All right, so this kind of goes through and gives you examples of again, missense mutations, nonsense mutations, silent mutations. All right, like I kind of went through before. All right, again, this kind of shows you the process of altering the reading frame. So it's kind of like if you think about the reading frame as a kind of a film strip. All right, so if you have a normal, a film strip, you have a normal film, and you have your frames. All right, and each frame is showing you a fraction of what's occurring in that film. All right, over seconds. All right, even sometimes even shorter than that. Well, what happens if you start cutting out portions of that, or you start adding stuff to it? Well, you start shifting and changing the film. All right, same thing happens in the DNA. All right, if you start removing some of the frames, you get a different part of the movie. You know, it's shortened. You know, you go from someone throwing a baseball to the ball being hit to the outfield, and you don't get the process of the batter swinging. All right, you miss stuff. All right, same thing with an insertion. It's like adding in a frame and adding in this new event in place of it. It could be something completely random that doesn't make any sense. But either way, it shifts the reading frame. It either shortens it or it elongates it. All right, and that can have an effect on the overall amino acid sequence of a protein. All right, so which of the following will have the most drastic effect on a cell? A point mutation, a frame shift mutation, a missense mutation, a nonsense mutation, or a silent mutation? All right, well, in this case, a nonsense mutation, because you start producing a non-functional protein. And if that protein is essential for a bacterial cell, then that cell's going to die. All right, now there are repair mechanisms in the DNA, or there are repair mechanisms for the DNA. All right, now you have proteins that can repair mistakes in the genetic code that occur as a result of DNA replication. All right, so we've already talked about very briefly that DNA polymerases have the ability to correct mistakes during replication, but even then, some of those mistakes aren't corrected. Right? You can have mutations that are due to exposure to chemicals, exposure to radiation that occur. And there has to be some way for the DNA to be repaired. All right, because if those mutations are passed on to the next bacterial cell, they're probably not going to be beneficial mutations. All right, so the two types of DNA repair mechanisms, we're going to talk about UV damage repair and excision repair, all right, or nucleotide excision repair. All right, so looking at the light-dependent repair of UV DNA damage. All right, so this involves photoactivation. It involves an enzyme called photolyase. All right, so in the presence of light, all right, if you have, for instance, a thymine dimer, all right, so basically this section of the DNA, you have this nucleotide that's attached to an adjacent nucleotide in the same strand. Well, that's not normal. All right, it's normal for two different strands of DNA that are anti-parallel within the same DNA molecule to be held together through hydrogen bonding, through complementary base pairing. That's normal. But it's not normal for two nucleotides adjacent to one another in the same strand to be covalently linked by their bases. All right, so this is a thymine, all right, dimer. All right, a lot of times this is a result of UV radiation. All right, so what happens is you have this enzyme called photolyase. Photolyase comes in, corrects this mutation, all right, this diamine dimer, and it repairs it. Right now, you have excision repair. All right, this is dark. This is labeled as the light independent repair mechanism. All right, so here you have excision repair, or nucleotide excision repair. You have the same mutation here, okay? What happens is that nucleotide, those nucleotides that have the mutation in it, the nucleotide gets cut out. All right, as it gets cut out, you have the DNA polymerase come in, it corrects the mistake. As you can see here, you no longer have the thymine dimer. And then this fragment of a nucleotide is removed. You have ligase comes in and glues this gap together, and the damage is repaired. All right, so looking at excision repair. All right, so basically you have, sometimes it may not be a thymine dimer. It may be you have the incorrect base. Right, so this would be complementary. Wouldn't be complementary to this. All right, and this wouldn't be complementary to that. All right, so you have the wrong bases. All right, so what happens is this part of the DNA molecule gets cut out. All right, those nucleotide bases get removed, and you get the correct ones added in. All right, so this would be complementary to this. This would be complementary to that strand. So you have complementary base pairs. So excision repair basically involves cutting and pasting. All right, cutting out the bad part, gluing, pasting in the proper bases that are complementary, and making sure they're glued in place.